What's going on, guys? So we're gonna go over the worksheet I gave you on Wednesday. We're gonna go over those answers. It was on one page is brain pop, and the other page was the different parts of the microscope. So we are going to do the brain pop side first. All right. So brain pop. Okay. What's going on, Jose? So for number one, which of the following is an example of a concave structure? So that's going to be B. All right. What's going on, Anthony? How are you, Brian? I'm doing good, Anthony. Yeah. Number two, why does a microscope stage have a small hole in it? So this allows you, when you're looking at a microscope, it allows the light to pass through. So number two is going to be C. Why are specimens usually very thin slices of material? Choose the best answer. So if we were going to be doing a lab in the classroom, one of the labs we would have done is the onion lab. So I would have cut it into really thin slices so you're able to actually have the light pass through so you're able to see your specimen. So for number three, it's going to be A. All right. The thinness allows light to pass through, which makes the specimen visible. And it also, since we're using an onion, okay, what else would you see in that? If you're looking at an onion, right, and let's say this is the actual field of view, you would get something like this. And why would all those little onion things look something like this? What are all these little circles in those squares? You guys know? So if you were looking at the microscope and you had all these little squares, okay, these are all the different cell walls. All right. And then inside of it, those little circles, that's your nucleus. Okay. So when we're looking in the microscope, you would actually be able to see the cell walls of the onion, because remember, an onion is a plant, and then it still has the nucleus, and then you would even be able to see these giant circles, which would be your vacuoles, okay? Because remember, plant cells have larger vacuoles than human cells. All right. So let's go to number four. Number four, which part of a microscope forms an enlarged image of a specimen? So those are going to be the objective lenses, okay? It's like a magnifying glass. There's three of them on the microscope. So number four is going to be D. Number five, if the specimen appears extremely blurry, what should you do? Choose the best answer. So for number five, it's extremely blurry. All right. It's a little bit hard because you want to think you're going to use the fine one. But if it's extremely blurry, you want to use the coarse one. Okay. The coarse one raises up and down the scale. And that's going to help you really get to see your specimen. And then the fine objective knob is going to get it really perfect. Okay. So it's going to fine tune. So five is going to be B. Number six, slides are made. Materials like plastic or glass. What does translucent mean? It means that light can pass through it, okay? So those slides that you would get are made out of glass, so you can actually see through it, compared to if it was dark, light's not going to pass through it, so you're not going to be able to see your specimen. And when I'm talking about specimen, I'm talking about what we're putting on the slide, okay? Number seven has a diagram, and it's asking you what the two arrows are pointing at and what about them. They are strong and sturdy, okay? So when you're picking up a microscope, you're always going to grab it from the base, and then you're also going to grab it from the arm and lift it up, okay? Number eight. Why is Anton considered the father of microscopic? Basically, he was the best microscope maker of his time. Okay, he was able to build all these good microscopes. Number nine. Why can electron microscopes magnify smaller objects? Because the wavelengths are smaller than visible light wavelengths. Okay, so number 9 is C, and that was right in the video. And then what can you infer about scanning probe microscopes? They are a relatively recent invention. Okay, so they're still recent. There's a lot of different microscopes. The one in number 10, they're still pretty recent to today. All right, so that was the one side for this. Now we're going to go to this side, okay, where we're actually labeling. So I'm going to keep this up for a second, okay? Remember, you can always pause the video and check your work. But now we're going to go on to the microscope side. All right, so remember, I actually labeled it 1 to 11, so we're just going to go right down, okay? So number 1, all right, it's pointing at the top of the microscope. So that is the eyepiece. That's where you're putting your eye, okay? So eyepiece. All right, that's the very top. That's where you're putting your eye on. That's why it's called the eyepiece. Now, number 2, going down, is connecting the eyepiece to the rest of the microscope. So that's actually called the arm, okay? It might be the neck, but we call it the arm for this, okay? Christian, I'll let you know. I'll give you a private message after this, okay? To show you your grade. All right, so number 3, okay? If you look at it, it's pointing at the black part of the microscope, okay? If you look really closely, it's actually looking at those metal parts on top of it. So those are actually called the clips, all right? So the clips actually hold what you're looking at on the base so it doesn't move, okay? So the clips are where the slide goes into it. All right, so now we're going to look at, there's 2 circles or knobs on the side of it. You have your coarse and your fine. Now the coarse one is always the larger one. So since number 4 is pointing at the larger one, that's going to be your coarse adjustment knob, okay? Coarse adjustment knob. Now the next one is the smaller adjustment knob. So that's going to be the fine adjustment knob, okay? The coarse one, that's the one that really moves the stage up and down, okay? And then the fine adjustment knob just fine tunes it so you can really see what you're looking at. Okay? All right, so number 6. Number 6 points at the top of the microscope again on the other side. Now, this is the rotating eyepiece, okay? Or nose piece. So the rotating nose piece, that's able to, at the top, you have 3 different objective lenses, okay? And this basically rotates. So you will either look at one of them, number 2, or number 3. And each one of those lenses are a different magnification, okay? And because they're moving it, so the nose piece actually controls the objectives, all right? And that's the actual magnification of it. Number 8 is pointing at the black stage, so that's actually just the stage. Number 9 is the condenser. That controls the light source, all right? So number 9 is condenser. It basically shows how much light's going to go in and out of it, okay? Now, since the condenser is right above something else, number 10 is the actual light source. And then the very bottom of our microscope, which I'm going to put over here, is the base. Okay? So that's all I wanted to go through for today. If you have any questions, let me know. But remember, you can also look at the PowerPoint. And then after this, I'm going to basically put out what the test would look like. Again, it's not going to be a real test, but I just want to see what you guys know if you were asked, the regular science questions that we would have done in the classroom, okay? So over the weekend, I want you to try the best you can, and then we'll go over it on Monday, okay? And then after Monday, we're going to get into the different body systems, and we're going to start with the skeletal, all right, and muscle system, okay? And then we're going to go through all the different body parts, all right? Does anyone have any questions on this? Yeah, I just said I'm gonna give you an assignment for the weekend, okay? It's gonna be due Monday. I'm gonna upload it right now after this video, okay? We'll go over it on Monday, and then we will be done with microscopes. I know we couldn't really do any of the labs, but then we're gonna get into body systems, okay? All right, guys, I'll see you later.